What the hell is the rhizophagy cycle? Well, in this video, we're going to get into it. You're here with Mark Boutwell on Perfect Gardens TV. Ladies and gentlemen, this is the video you're not going to want to miss. James White is one of my heroes in the soil microbiology direction. He is literally one of the gentlemen that is leading the charge and is cultivating the minds of the people that are going to go on and teach the farmers that are going to inherit the earth with healthy growing practices. Without further ado, let's go ahead and get into it. Please remember to like, share, and subscribe. If you haven't checked out our monthly membership, I highly recommend to do so. For only $2.99, you get access to over 1,570 photos, 162 videos, 10 files, 15 audio files, 562 shared links, and 86 members willing and ready to assist you through your growing practice. Make sure to hit the join button on the bottom of every video.
Okay, so basically what I'm going to talk about are how plants are internalizing, and I'll say soil microbes, because all of these microbes that they internalize come from the soil initially, and they adapt some of them so that they can utilize them better. They'll select for the ones that can use the best, and they'll put those onto the seeds of the plant so that the seeds, when the seeds germinate, they'll have these bacterial endophytes in them, or fungal endophytes. Okay, so endophyte, what is an endophyte? Endophyte is any microbe, fungus or bacterium that goes inside tissues of plants. And for example, in grasses, forest grasses or turf grasses like fescue grass or ryegrass, you have these fungal endophytes. You also have lots of bacterial endophytes in there, but you have these fungal endophytes that are well known, and people may know the term endophyte because of these fungi that go in these grasses. And this is a fungus, and you can see it here stained blue. That's the hypha of the fungus, and it goes between the cells. In this case, it's what we call intercellular, going between the cells. But it goes in the grass, you don't even know it's there. You got to have a microscope to look at it. Okay, endo, meaning inside, phyte, meaning plant, endophyte, any fungus or bacterium that goes inside of.
So that blue is actually traveling through the cell, correct?
It's actually, if you look in the background, look at this, you can see this little, these, where my arrow is going around like that. Those are the cells of the plant. So that blue, if you look at it, that blue is the hypha of the fungus that is on the surface of the cell. So these endophytes go into the tissues of the plant, but they stay between the cells in what we call the intercellular space, right? But we have other endophytes that actually will go inside the cells in the intracellular space. A lot of these bacteria do that, and that's what I'll be mostly talking about now, are these endophytic bacteria. Basically, these bacteria, plants suck them into their tissues. They suck them in from the soil, and they'll degrade some of them, and some of them they'll then train to work as endophytes on the plant, and they'll keep them in there. And you see them, for example, in roots, and I'll talk about this process, but you can't see them. In fact, people have not seen them typically previous to, say, 2010 or so, because they have to be stained. Otherwise, you can't see what they are. And so we developed some stains to use them and to view them, actually. And you can see all these stains here, and I'll be showing images of this DAB, NBT, ammonium molybdate, aniline blue. Basically, these stains stain substances that are produced by the bacteria or produced by the plant in response to the bacteria, so we can see them inside and you need a microscope. Okay, so you've
only stained what you know is there, correct? Like you can only what you know is there, you can identify, put a stain to it, and then identify. You can't just like put something out there randomly and see every different types of things, possibilities they're creating. Is that correct?
No, we can take any of these stains. Like, for example, a lot of these endophytes are producing ethylene. So we could take that ethylene stain, one of the ethylene stains, we have multiple ones we could use. We can put them on the plant, and anywhere these microbes are producing ethylene, we can see the microbes, right? So, I mean, we could use it to screen plants, and that's what we did really more recently. We're looking at leaves, trichomes and trichome symbiosis, these bacteria that go into trichomes, and I'll talk about that. But this is an example of one of these plants. This is from the desert island of Bonaire. I did some work there. It's nice to go to these places to do. This is one of the islands in the Dutch Antilles, but it's a desert island. People like to go there for diving, but also they have these cacti all over this island. And if you look at the cactus fruits, you can see that just like the prickly pear that we have in North America, but those fruits take the seeds out, and seeds like that, and then germinate those, and you get a seedling like this. And then if you look at the root hairs of this seedling, which is what I'm showing right here, you can show those little root hairs, and you can see the little dark structures inside those hairs. Those are the bacteria.
Wow.
That the plant took in. So these plants are sucking in these microbes out of the soil, but they're also putting them on seed so that they can then give it to the seedling, and the seedling will have these microbes for the benefit of their growth. And I'll talk about how that works, but it's really, really interesting. So plants are consuming microbes or absorbing microbes from soils. This shows a root hair again. You can see these bacteria here, and you can see the pairs of bacteria. And these roots just get filled with microbes. That's just one example of a plant, but all plants, every vascular plant, all of the higher plants, we don't know how far back this system goes where plants are using microbes, but more than likely, even the ancestors of vascular plants even non-vascular plants possibly, were taking in these microbes to get nutrients from them. We don't know, but this may be a basic plant thing. And crop plants like, here's one, cannabis, right? Cannabis. I have a student, April Michi, trying to work on cannabis, but it's really hard when it's illegal or when it's not fully legal yet, which is what we have here, not fully legal yet. But if you look at the cannabis seeds, they actually see the seeds here, they have bacteria all over them, and internal too. So they vector these microbes on the surface of seeds and also inside, and they will suck them out of the soil as well. And this shows a root hair from cannabis. See all these little brown dots in there? Those are bacteria that the hemp took in. To the right, you see a bigger magnification. See the spherical structures? Those are the bacteria that cannabis took in into its structure. Tomato, you think tomato inside the tomato fruit has got to be clean, right? Far from true. There are microbes being vectored in those seeds and around those seeds, so that when those seeds germinate, they'll have bacteria. They go into the embryo, into the seedling. If you look, this is one of the bacteria we got from tomato. This is something called Micrococcus luteus. And we like it because it has these little packets of cells, these little tetrads, they're called. So they're very distinctive. So you can see them when they go inside the plant. And as it turns out, when a seed germinates, the root comes out, and then those bacteria on that seed or from the soil will then colonize the root tip. And this orange area is where the microbes are colonizing and going into the cells. And then orange actually is ethylene that's produced by those microbes. You can see also right there at the very tip to the right, the purple color, that's where the bacteria are going in.
Could I ask a quick question? Specifically, what is ethylene? And specifically, what is it?
It's a good question. Ethylene is a plant hormone, and ethylene is used by plants to modulate or to control fruit ripening, for example, but also tissue growth. And it's a hormone that will cause cells to elongate and will also cause maturation, right, in fruits.
And the bacteria produces it, correct?
The bacteria are producing it. And, this process, I would say it's a way that bacteria can control plants, but in a very negative or very limited way, because bacteria produce ethylene, and that causes nutrients to be released from the plant cell. So it's a way that bacteria can get sugar coming out of the plant. So it's kind of a communication, right? It's a communication tool. It's like the microbe saying, hey, give me sugar, put ethylene out, give me sugar, give me photosynthate. And then it leaks out, and then they can get some of that.
In one of your previous videos, you're talking about how when the bacteria comes in, it's stripped, and I don't want to get too far ahead. The bacteria gets stripped, and the plants are producing sugars, and I don't want to call ATP a sugar. And sometimes I want to call adenosine the sugar, and then phosphates the minerals. But And then I kind of heard someone say that the sugars are manipulated and changed and then attached to the bacteria and then recoded and sent back out through the root hairs. I don't know if I said that correctly, but my question is when are the plants communicating to the bacteria at that moment to tell them what they're malnourished on through that ethylene communication transfer connection?
That's a really good question. The plants are, I'm going to put it a different way.
Okay, please.
The plants are cultivating the bacteria that they need in order to get the nutrients that they need. So the plants alter the microbes that they cultivate, right? And how they cultivate them. How do they do that? They do that by changing what nutrients they're putting out. Okay, for example, if the plants are looking for phosphorus and potassium and some other nutrients, that's what they need, then they may have some nitrogen in addition to sugars in their exudates, in their root exudates. And that allows, because the nitrogen is out there, that allows these the bacteria and the microbes that don't fix nitrogen, allows them to grow and then to acquire these other nutrients that the plant needs. And then they'll take those microbes in, they'll absorb those microbes and get those nutrients from them, or they'll absorb those nutrients from the water, the solubilized component in the soil. Okay, so in other words, they are giving the nutrients that enable them to get these other nutrients, right? If, for example, they are short of nitrogen, they will continue to put sugars in their exudates, but they will withhold any amino acids. And that has nitrogen in it, the amino acids. And that forces the microbes to fix nitrogen. And then they will take those microbes that are fixing nitrogen and take those into their tissues. That
sounds like an osmotic effect. If they're, does that sound correct? Like if they're if they're not allowing the amino acids to have, amino acids are nitrogen, carbon, oxygen, sulfur, and I'm missing one of them, I'm correct. But if they're restricting it, then is that creating like an osmotic effect? So when they're traveling around, it's sucking in to find balance?
It's not really an osmotic effect. What they're doing is they're managing the population of bacteria in the soil. And they are, by withholding amino acids from the exudates, they're forcing those bacteria to fix nitrogen. And they're selecting those nitrogen fixers in the environment, right? And then they can then get the nitrogen from those nitrogen fixers. So what they're doing is actually Jeff Lowenfeld said it nicely. Plants are ranching microbes, and they're cultivating the microbes that they need, growing the microbes they need, and then they're taking them in and taking nutrients from them. And so they are cultivating the microbes they need depending on what nutrients they need. But it's a little different than mechanism that you suggested. It's a little bit more microbe population management approach rather than an osmotic nutrient absorption.
I appreciate that
approach. Yeah, no, you're welcome. I mean, it requires a little bit different thinking about it, right? You think about it a little differently. Yeah, Dave.
Going back to the ethylene that the bacteria is producing, is that the similar process with auxins, where like certain products like kelp meal or kelp products, seaweed products have shown to have auxins, root enhancements, and these hormones? Is there a similar process? Is it ultimately then they end up making ethylene from that, or is that a totally different process with auxins?
So it's a different process, but some of these microbes can also make auxin. And it's been hypothesized for a long time that these microbes that can make auxin are stimulating plant growth and development. Okay, we don't see that in the studies that we do. We're not staining for auxin. So we're instead, looking at these bacterial plant cell interactions, and we have not yet developed a way that we can visualize auxin in that exchange. So there could be auxin produced by some of these microbes. We just don't know. I mean, we're putting together the puzzle based on, the studies that we're doing. And we have not, gone into the literature and just fished out and said, well, this must be here. Well, we don't assume that. Instead, our work led to ethylene as the key component here. anyways, those are good questions. And you can see, look here, this is a root. See root cells here, and you can see those little tetrads inside those cells. So this is just so that you can get a close look at the bacteria inside the cells. And this is in the area that these bacteria are going in. Okay, but all these are soil microbes. They're initially from the soil. So plants need healthy soils in order to acquire these microbes from those soils. These can be fungi. And when fungi are taken in, they actually, mycologists gave them a new name. They call them mycosomes, little fungus bodies. But the same thing happens to them when they go in, except here, no fungi can fix nitrogen. So there's no nitrogen fixation or anything like that happening with fungi. But instead, plants are affected in a similar way. This work was published in PLOS One. It's one of the open access journals back in 2014 by Peter Atzat and Michael Whitesides. Okay, so this is not from my lab, but from other labs, other work. Really interesting study. This process of root internalizing microbes was first discovered in 2010 by an Australian group, and Queensland, Australia. And what they did is they found yeasts and they found bacteria were going into root cells. And so they then tagged the bacterium with the green fluorescent protein, which you can then put into the DNA of the bacterium or the yeast. And then you could look under a microscope and you can see wherever those bacteria go in the cells because they'll fluoresce. So this is something from their paper. And you can see a root hair here at E. See a root hair and see the little dots in there. Those are the yeasts in there. And their paper here, they titled "Turning the Table: Plants Consume Microbes as a Source of Nutrients." That was their title. And they named this process rhizophagy, rhizo for root, phagy for eating, rhizophagy or root eating. And then we've been studying it since then. And what we have seen is this is actually a cyclic process. And that is that plants are attracting these bacteria from the soils to the root tips using exudates. And then they will take them into the root cells and they'll hit them with superoxide, and that will take their cell walls off. And this is what you referred to, Mark, before. It oxidize off their cell walls off of these bacteria, and they'll become these naked protoplasts inside the cells, and then they'll continue to hit them with superoxide, reactive oxygen, and then they will continue to get nutrients out of them. They'll replicate the ones that survive. The survivors then will produce enough ethylene to cause these root hairs to elongate. And so then you have root hairs forming, and as those root hairs form, the plants squirt the bacteria back out into the soil. They squirt is kind of non-scientific word, but they eject them. Every time there's a growth spurt, they eject some bacteria, they grow again. And every time there's a growth spurt, it's because the microbes are producing ethylene. So if we remove all these microbes, you have no root hair formation. Or if we suppress ethylene using some other method to do that, we can do that. And then you get no root hairs forming. So plants only have root hairs because they have bacteria in those root cells. Okay, so they're ejected out. Okay, and then as you mentioned, Mark, they will reform their cell walls out here. And it is true that some sugars will go out with the tips of the hairs and give them some nutrients so that they have the nutrients they need, the energy they need to reform their cell walls. Then they'll go back out into the soil, they'll acquire nutrients, and then later on, they'll be attracted back to the root tips that are growing. And so you have this cycle where bacteria acquire nutrients in the soil, and then they are internalized into the root cells where the nutrients are extracted from them, and then they go back. And so it's just a continual cycle like this. We think that some bacteria are fully degraded, and they don't really participate in the cycle, they're just consumed. Other bacteria do cycle, and the ones that cycle are the ones that plants cultivate in the rhizosphere, this area outside the root. So
sorry, go ahead, Dave.
I was just going to ask you guys found exit points through these little tips. Have you guys found, like, how are the bacteria actually entering? Like, are they maybe eating or going through that cellular structure, or are there access points, or where are you guys at with
How
that?
they get in.
Or is it through the tip?
It's at the root tip, but the walls are thin at the root tip. They're soft, right? And that's the spot that the plants are putting out these exudates to cultivate the microbes. Okay, so we do not know exactly how they go through that soft cell wall. One idea proposed by the Queensland, Australia group is that it is cell eating or phagocytosis, that the cell actually can get them somehow. It's hard to see how that happens with a soft wall there, but another idea is that the microbes will degrade their way in. And once they degrade their way in, because these microbes all have cellulases for the wall that's made of cellulose, so they can get in there, pectinases and stuff like that, so they can get in. But once they're in, then they're at the mercy of the root cell and superoxide, and they can't produce any enzymes. Then once they're in, they can't do anything.
They're kind of like a parasite. They're kind of like a parasite at that point.
They're
Needing a
more
host.
well on the outside. They're like a parasite. They could be a pathogen, but once they're in, they're not. Once they're in, they're a captive. They're a captive because the plant with superoxide will oxidize any proteins they have, any cell walls that they make, and that prevents them from getting out or prevents them from doing any damage because they can't make any enzymes, right? The plant is zapping all of it.
And superoxide, is that O3?
That's O with an extra
with a negative? Oh, it's O with
negative.
negative.
O
negative.
Yeah, it's highly potent. It's highly potent form of reactive oxygen, but it's very short-lived. So it's a very precise type of reactive oxygen. Yeah, you could have other forms, but that last longer, like hydrogen peroxide, but they're much weaker, right? Superoxide is very short-lived, but very, very potent. So it's probably the logical molecule to use. This whole process, by the way, well, I won't go into evolutionary cycle, but of course it's tied to getting oxygen in the atmosphere, right?
And you can
oxygen levels
stem into another conversation for a minute or two. It's okay. This is kind of how we do our dialogue. We talk until we don't know, right? They're like, I didn't think about that before. So the next one I had a theory around. It's just me just listening to you guys and then interpreting it. When you say some of them get dissolved completely and some of them get in a sense, a plant communicates, something attaches to it and gets resent back out. My question is the ones that get dissolved, are those the bacteria that have completed their cycle and have returned back to the plant with the nutrients that they have requested or required?
No, those would be the ones that cycle, right, all around. Those are the ones that keep cycling, that survive and set up a sustainable symbiosis with the plant. The ones that get fully dissolved would be ones that are not very oxidatively resistant. And what would happen is
pathogenic bacteria.
They might be pathogenic. They might be just other microbes that like sugars that the plant's secreting, and they get caught up in this process going in with these other bacteria. Then when the superoxide hits, they're not able to tolerate it, and then they're fully degraded, right? Superoxide, they cannot resist the superoxide. Their protoplasts are fully bombarded by superoxide. Now, we think, right? We don't have a lot of experience with those, because those types that are highly that don't cycle, because we're studying the ones on seeds, and the plants put on their seeds the ones that work in the rhizophagy cycle and as endophytes in the plant. And to do that, you have to be oxidatively resistant to some extent, right? You can't be fully degraded when the plant puts superoxide on you. Okay, so there's a lot yet that we don't understand about the process and that we have to learn. Okay, so the process, all of this rhizophagy cycle happens at root tips. And so you can see here, this is a grass. Grasses have a lot of root tips. You can see all the little lines all over these roots. Those are the tips of roots where rhizophagy cycle would be happening. So grasses do this a lot. And so they'd be getting a lot of nutrients from bacteria in soil through the rhizophagy cycle. But all plants do it. All plants do it in the roots, and they take it in, just some more than others. This is a grass root, and you can see the root tip here, and you can see the kind of a blue haze out there, the blue cloud around it. Those are the bacteria being cultivated in the zone of exudates, where the plant is cultivating those bacteria with exudates or sugars, right? So plants are smart. You know, this is one way that they're getting these back. They're controlling this process. Plants are in control of the process of getting these bacteria to come and then taking them in, and they'll feed these bacteria. In order to do this, they'll give them as much as 40% of the photosynthesis that they make, of the sugars they make. They're giving them to these microbes in order to get these other nutrients, these other soil nutrients from the microbes. So plants are in charge of this process. And to a plant, photosynthate is cheap, but these other nutrients are hard to get, you know, the phosphorus and the potassium.
So the picture shown, would this be the process in where they're taking in and potentially ejecting, like the or where they're actually coming out? Like, what part of the root is
this? This is the tip, the root tip here of a branch root. So you can see it coming out of this bigger root, right? And this is where they're taking them in. Further along in this root, once it gets bigger, they would be coming out on the root hairs, and I'll show you that. Okay. So you see what I'm talking about.
And not to be tomato, right? Are they sugars, or are they amino acids and peptides?
They are a mix of nutrients, mostly sugars. Mostly sugars. Sucrose,
glucose, those types of sugars, or are they
yes
evolved forms of sugars? Like
they're sugars that microbes might like.
They are actually sugars.
They are actually sugars, but they will also put out amino acids in certain circumstances, and also organic acids in other circumstances. They're essentially chemicals that the plant can control these microbes with. You know, that's the important thing.
Three things: amino acids, organic acids, and sugars are being released to control the microbiology in the soil.
Yes.
Interesting. Okay.
Yes. Yeah. Plants are in control. You know, plants are ranching or farming microbes, soil microbes. You know, it's a principle, right? So this is a root, and you can see the root cell. This is the epidermis. You see those little spheres in there? Those are the bacteria. These are those protoplast stages. And you can see they're replicating in there. Okay, so the plant is actually replicating these protoplasts, bacterial protoplasts inside the cells. And let's see. Now this is one of my students, one of my graduate students, Celeste Song, and she actually did one of these tagging studies. And this is where she took a Pseudomonas and tagged it with a fluorescent stain called M. cherry, and then put it under the confocal microscope and examined it. And here you can see her scanning the image through these cells of what was this plant? I don't know, clover plants, I think. These are the root cap cells, and you can see the red things there. Those are the bacteria inside those cells, inside those plant cells. Wow. So yeah, no, you can see, and see that's labeled. That's labeled. So she knows the bacterium, Pseudomonas, and then she put this tag on it, and then she put it in, and now you can see it in the plant. You know, you can do this and see it in any parts of the plant. So we know where these microbes go because we can put a little label on them and then track them in the plant. So as it turns out, okay, most of these bacteria are, at least in the rhizophagy cycle, are staying just underneath the cell wall. Okay, so this is a drawing of that. This gray is the cell wall. And you can see the cell in the middle here. You see the little line that my arrow's going around. That's the plasma membrane of the root cell, right? And then you see the membrane. It's out there between that plasma membrane and the cell wall. So it's in this little space called the periplasmic space. So it's not actually in the membrane or in the cell itself. It just stays out there. And it's in that little space that the plant puts superoxide to dissolve the walls and make those bacteria leak nutrients. This whole thing, by the way, the plant, this is part of the plant control again. The plant is moving these bacteria around like this in a process we call cyclosis. So it's circulating like this around and around and around like this. And what that does is, okay, so the plant's hitting these microbes with superoxide and then it's moving it. Those bacteria cannot, they can't, they're in total control. They're being constantly moved and the plant won't let them reform their cell walls or produce enzymes or whatever it is. They can't, the plant is moving them. The movement enables the plant to break these microbes up into smaller and smaller little protoplasts and replicate them, so the plant can replicate them, but the microbes themselves can't then suddenly become pathogenic or something. They're under control in this process. So
the paraplasmic area, this is kind of like, it's kind of like a waiting room and a conditioning room for the bacteria and what the plant was going to want to ultimately do with it.
Okay, not a waiting room, but a processing room.
Right.
Conditioning room.
Yeah.
Conditioning room is better.
Yeah.
Where these microbes are being processed right in there and nutrients are being extracted. So
how long have we known that space was there?
So crazy.
Oh, we have known about that space for a long time, but we have not known that there are bacteria in that space. And the reason is because bacteria are not unless it's a disease, they're not supposed to be inside plant cells, right? Or animal cells, So we don't expect to see them there. So we don't interpret them as that. In fact, when people see them, they say, oh, there's some extracellular vesicles, right? Extracellular vesicles. Instead, they don't interpret them as bacteria or microbes, but instead some kind of extracellular vesicle or a vesicle outside of the cell. So whatever they are, whatever that means. We have images of electron microscopy where they see these things, they show these things, and they interpret them as vesicles. So it's a phenomenon.
Wow.
That you don't see something until you first see it.
What's with the variances in superoxides between those paraplasmic spaces where some have 3, some have 1? Is that just the variances of what it's trying to extract from that bacteria?
No, that's just artistic rendition variation.
Okay.
We don't know how much they differ. Yeah. But when I made this diagram, I just had some of these 2 O minuses or O minus things. And I just stuck them.
Got it.
No, that's cool.
Yeah,
that's cool.
That's what it is. So the enzyme on the plant cell,
you didn't go to school to paint the Mona Lisa, right?
Believe it or not, as a mycologist in the early days of mycology, I illustrated a lot of fungi. And yeah, I'm trained, believe it or not, as a botanical artist.
Wow. That's
believe
cool.
it or not.
That's cool. That is cool.
We all had to back in those days. We all had to. Now people don't do it, but back in the 70s when I was in the early 80s when I was training as a mycologist, we learned to do that.
It makes sense, right? You have to transcribe what you see in a book because the internet wasn't there. So how do you transfer your information to the rest of the world at their discovery?
And photography wasn't as good as it is now too. We didn't have digital photography. Everything was like film photography. So everything was black and white. So you had to use that. And illustrations in botany, people illustrate things to show the points they want to show, right? So you had to be a botanical illustrator or fungal illustrator in order to show the features that you want to show. You can't take pictures of everything and show it because they don't show up good in pictures. Yeah, so you do have to be an artist. Yes. Yeah, good question. Really good question.
That previous slide that you just left, was that the actual driving force of the oxide, superoxide?
It is. This is the enzyme that's in the plasma membrane of plant cells that produces superoxide. This is one it's an enzyme called NADPH oxidases, nicotinamide adenine dinucleotide phosphate oxidase, right? That's the enzyme that produces superoxide. And what it will do is it will take oxygen from the air and will make superoxide. And that superoxide then can zap the.
What's the cylinders that look like attached with iron or right in the center? What is that part?
Oh, that is, yeah, those are cofactors in NADPH. It has iron in it. Yeah, that's a good observation. That iron.
And then is that what form of iron? Is that oxidized iron or is it non-oxidized? Like, does that matter? Because I know the plant can take up different forms of iron, but not necessarily can't use it, which I'm still trying to grasp. But so is there a specific iron in that case?
So I don't care to answer the question specifically the way you asked it because I don't want to get it wrong. Right? I don't want to get it wrong. It is a form of iron that is in one of those oxidative states that you talked about, and which one I don't know, that functions as a cofactor in this NADPH oxidase enzyme, also called NOXes. These are important enzymes. We know this takes molecular oxygen, right? So this whole process could not have evolved prior to getting oxygen in the atmosphere. And of course, that's when plants began to evolve. And so that oxygenation in the atmosphere is when plants began to evolve on land, and when this whole process, rhizophagy cycle, probably started way back in the evolution of plants. Yeah.
So if people are potentially having or showing signs of, let's say, an iron deficiency, in my mind, that system would be affected as well, where it's not super oxidizing maybe as efficiently as it could because of the lack of iron, or
and sulfur,
kind of like my mind's
and
going
sulfur
to.
too. It connected to the sulfur because sulfur with the iron always have a relationship, right?
Yeah, I mean, it connects with other nutrients. In terms of deficiency, these nutrients are being brought by the bacteria. I mean, iron is one of the main ones brought by bacteria. So sulfur too can be brought by these bacteria. So the process can support itself in that respect. They can get this from the bacteria to then utilize in the plant cells to get more nutrients from the bacteria, right? So yeah, deficiency is there. Plants do have these deficiencies, and they precisely, I actually studied a little bit how they use nutrients, exudates, in order to get bacteria to produce iron. Oh, you know what it is when they want iron? This is another interesting thing. When they want iron, what plants will do is, another thing that they'll do is they will secrete amino acids in their exudates. And what that does is it will favor these other bacteria that are not nitrogen fixers. And the ones that fix nitrogen use a lot of iron. And so they'll take up the iron from the plant, and the plant will have a deficiency of it. But if they put amino acids out there, you don't get nitrogen fixers absorbing the iron, and instead the plant can take that iron. So you can see how they can also utilize exudates in order to get more iron, right? When they're iron deficient. So yeah, I don't know how much sense that made, but it's kind of relevant to this deficiency idea that you talked about. So, okay, this is just a picture. It shows bacterial rods. This is with their cell walls. That's what they look like in the soil. When they go in to the root cell, they get hit with superoxide and they lose their walls. And now they're these spherical things. You can see this is to the right. That's the spherical things. Those are the protoplasts. They've known about these a long time. Not so much. Even in plant cells, they knew about these L forms. They called them L forms. But it was discovered at the Lister Institute, and that's what the L stands for, Lister forms. These are bacteria without their cell walls. And back in the 1970s, people were seeing these L forms in plants, and they were talking about them, but they didn't connect rhizophagy cycle or nutrient acquisition process with this, so that connection wasn't made. But they were known to be endophytic or symbiotic forms of bacteria. if you take away all the microbes from plants, the plants don't develop correctly. And one thing that you see is that the roots don't form correctly. Oftentimes the roots will lose the gravitropic response. Normal roots go down to the soil like that. But if you take away all the microbes, they won't. They'll stay on the surface, or in some cases, they'll just go into the air. So they lose that gravitropic response. They need those microbes for that. Why exactly, we don't know. They also will not form root hairs. You don't have bacteria, there's no hair forming. We talked about that. And that's a direct effect of those microbes. So, I mean, to show that, here's a grass. This is a Bermuda grass where we removed all the microbes. And you can see these are parts of the root there of the seedling. You see there's no hairs forming on those. But if in the same experiment, we take it and put a Pseudomonas, a bacterium on it, and now you can see hairs forming immediately. And and you know why? Okay, here you actually see an older bit of the root. You can see the little brown dots in there. Those are bacteria in there, and you can see the brown inside these root hairs, and those are all the bacteria inside those root hairs. Plants. Root hairs
have some plants evolved maybe in environments with less amounts of bacteria. Like when you were talking about the root structures going down, and then if they don't have what it takes, they tend to stay at the surface. And right away, my mind started thinking about pine trees. Like pine trees seem to always have shallow roots of what I've seen, or certain spruces, like, you know, where you see them in the forest floor and they fell over by a wind, and, you know, it's a big pie of roots, you know, where they don't necessarily have that deep taproot. Was that maybe an environmental evolvement due to lack of certain bacteria? But at the same time, the forest floors are filled with a constant carbon cycle, it seems. So
in the last 50 years, most of our forest has been cut down and recommercialized and replanted. So depending upon what type of, you know, our humic layers have been destroyed through that process, and then replanting, are they even considering microbiology? And obviously it's going to come back, but I'm just throwing that out there too. I want that's a very interesting. I've seen the same thing. Huge root system this way,
right.
Not a large root system downward. And is the root system just snapping? I don't know. Do you know anything about that, James?
Yeah, I do. So, I mean, it's really a good point. But it puts the plant in control again, right? Puts the plant in control. So what happens in first of all, we're looking at I have some collaborators in Germany, a lady named Judy Simon actually, and they're looking at rhizophagy cycle in forest trees, and also bacterial endosymbionts in the forest trees as a source of nutrients. But plants, trees in specific, and many other plants have evolved associations with fungi, mycorrhizae, right? Ectomycorrhizae, for example, in these pines and stuff, that really replaces all of this kind of root rhizophagy activity cycle, right? Because instead what happens is these fungi, ectomycorrhizal fungi, will coat these roots in all parts of the root. And then instead of having root hairs go out there, instead of processing those microbes the way I described, these fungi will go out there and they will then acquire those nutrients and get those back to the plant. But now there is a big unknown, and that is there a connection between these soil fungi, mycorrhizal fungi, and, for example, other microbes that may be, for example, fixing nitrogen, right? And there may be, because we know that some fungi will have bacteria that go into them, but it's a big unknown, really. It's a big void there. So we don't know if bacteria are connected to those mycorrhizae.
My question was tell me if I'm wrong on this. And this is just my theory of just reading and stuff. The fungi are what holds on to the sulfur, correct? And it's also why we see these blooms after rain falls, is we see the mushrooms pop. And we were talking about the relationship with the iron and sulfur. are there bacteria that deliver and grab sulfur and bring it back to the plant, or is that the connection? Do the fungi hold on to the sulfur? The bacteria get the iron, and then there is an iron sulfur relationship that, with the change of chemical structure, that then are allowed those molecules to be brought back to the plants.
Well, there are a lot of chemical interactions that are happening, and I cannot break out any particular ones that relate to sulfur and iron. I could shed any more light on your question, maybe answer this better. All that I can say is that there are a lot of complex interactions, and that result in what nutrients the plants are acquiring. And I mean, even if you look at what I was talking about, the relationship between exudates and nutrient needs in plants, it's not an easy thing to decipher. It's complex, and there's a lot of work that needs to be done to understand exactly how plants are getting specific nutrients from these microbes. I mean, that's all I can say, is that there's a lot of unknown here.
No, thank you. I appreciate it.
Yeah, well, I hate to give an answer like that, because basically, you gave me a fairly specific question, but I can't really answer in a specific
I'm
way.
hungry to dig. That's what's so exciting about education. You don't know. I'm like, yes, all right, we get to research more. It's fun. Wait for more years, have some more videos with you.
Well, I mean, we're really excited about what we're finding now. And we're going to get to that pretty soon. The rhizophagy cycle is one aspect, but plants are absorbing these microbes into all parts of their tissues. This actually is a root hair, and you can see all the bacteria in it. They become full of these bacteria. And why is root hair growth specifically associated with bacteria there? Or maybe even be fungi in some cases, because they also are producing ethylene. Ethylene is the key. And this is another one of my graduate students, Ivy Chang, and she did some work that shows, you put the bacteria, you remove all the bacteria. This is a root over here to the left. There's no hairs. Put the bacteria on and you see their hairs. And if you look at the tips of these little hairs, little heads there, those are bacteria that are coming out of those cells. And why? It turns out that the microbes are producing ethylene. That's the theory. Ethylene, and we have experimental evidence that suggests that. We also know that the microbes are producing nitric oxide, or nitric oxide is being formed. And both of those, nitric oxide and ethylene, are hormones for the plant, and it will cause cell growth. But anyway, we have this, and those microbes accumulate. The picture to the left, you can see them, little black things, those are the bacteria in the root tip, the hair tip. So you could imagine that, those microbes there in the tip secreting ethylene that's causing that hair to continue to grow, to elongate. So okay, here's one of the plants from Bonaire. Again, that was a nice trip, getting multiple species studies happening there. But this is a sedge, and they grow basically without soil, just in these little rock areas. And if you look at the root hair below, this is the sedge root hair, and you can see the red areas. Those are the bacteria, and they're in that periplasmic space. But look to the above.
Wow! Look at them travel. Look at them travel. That's amazing. How fast is this sped up? Is this
no, just slightly. This is about 11. Let's see, these move at about 11 micrometers per second. So
Holy crap, that is so fast.
Yeah, so this is what happens. This is what you see in a living root hair, right? But if you pull that plant out of the soil, this stops. Pull it out of the soil, put it on a slide and look at it, and you don't see this.
Wow.
So this is how plants in root hairs manage these microbes. This is
how fast do they come back? I think about transplanting now. How fast do they come back?
It would just be probably a day or two, something like that. There's actually articles about this. and specifically nitrogen fixation. Turns out nitrogen fixation appears to depend on this process in some plants. And if you stop the movement or you disturb plants, then nitrogen fixation stops. And we think it stops because in order to fix nitrogen, the plants have to be doing cyclosis in their tissues.
Do they stop because of the light, or do they stop because they're taken out of the soil?
Because they stop if you pull the root out. What happens is you move the root hairs and you break what's called the cytoskeleton, right? The cytoskeleton, the cell skeleton. The cytoskeleton is this internal skeleton in these cells that's made of microtubules and microfilaments. And these are little protein structures, right? And if you move the hair, it breaks, and then the cell can't move. It can't move, it can't do cyclosis. So it takes hours or days, depending on the conditions, for that cytoskeleton to get rebuilt. And then you get this movement again. Okay, so that's the short answer. The long answer, I guess.
No, thank you. That's awesome. That's awesome.
Yeah, no, it's cool. We don't think of plants as doing this, but plants are very active. And even a root hair is not just this little thing that's just sitting there absorbing nitrogen from the soil and other nutrients. It's active like this.
It's a very complex machine, man. God, look at all these processes.
It is a machine. It is a machine. It is a machine. There's a mechanism for it. A plant, I mean, and you said it, Dave, a plant is a microprocessing machine.
Yeah.
It's a microprocessing machine. Animals are not. You think of animals, we get the microbes into our gut. We don't put them all over our bodies. I mean, they are on our skin and so forth, but we try to limit them in our guts. They don't go in our cells like this.
Right. We always give Henry Ford the credit for, popularizing the industrial age and assembly line. But geez, man, these plants,
they do it. They really are a machine here for doing it. And it's really just really cool.
Microprocessor, just like Dave said, computers. Again, I was just like, oh, wow.
So this is one of these root hairs, and you can see these arrows are showing clusters of bacteria, because when the plant is doing this in their hair, it's jostling these bacteria around, and it's replicating them, cloning them, right? And so you can see probably this cluster here that to the lower arrow probably came from a single bacterium that the plant replicated and made a bunch of. And so now it's cloning these bacteria that work with it, and it's going to put them back out into the soil. It's cloning them, the good ones, right? The bad ones are fully degraded. They're not that useful probably, but these are the ones that work with it pretty well and that go inside and survive, and there's the nutrient transfer. Right?
So when cells regenerate, you're telling me they're also regenerating the bacteria that's in them as well?
They are.
Wow. Oh my God. That's amazing.
Bacteria. They're cultivating, they're ranching, they're farming, however you want to put it. But they're growing cells typically will have some of these bacteria in it.
They really are farming cells. They are cloning. They are cloning the bacteria. They are actually generating the bacteria. That is amazing. They're not just taking it up in the soil. They are creating it.
They are causing them to replicate inside, and they're replicating them. Yeah.
Wow.
No, it's absolutely right. They are farming these microbes. This is actually, look at this. This shows a root hair.
If we can get them to farm Bitcoin, holy crap. No, I'm just kidding. Let's keep going. I'm sorry.
You see the tip? You see the little clouds in the tip of this? Can you see that? The little
they're accumulating
a lot up there. In the tip. Okay, so that's what they do. They accumulate in this tip. And then that's from there where they secrete ethylene, and then they will cause that hair to elongate. And when they do that, they eject the microbes. This actually shows one of these, and you can see it's filled with microbes. It's fat with microbes, with bacteria. Some of these are red, some of them are clear, but there's still lots of microbes. Only the ones that touch the membrane are going to be stained red like this because that's where they get exposed to the superoxide and so forth. But the others that aren't touching the membrane are, we don't see them stained here, but they're filled. This hair actually was the actual membrane, the little cell of the hair is a little needle-shaped thing inside this big club-shaped root hair, right? So the cell itself, the plant cell itself is little. And all of this stuff out here, these are microbes in these hairs. And they're coming out. And you can see here to the left with its arrow is, these are all bacteria that have been ejected out. So they're pumping up, they're making a lot of these microbes and they're pumping them out the tips of those hairs. And so those are going back to the soil in more massive numbers. So the plant is really engineering this rhizosphere environment, these populations in the rhizosphere, putting out more of these microbes that it likes and it uses, right?
They always say if you want a large root system, you want phosphorus, but no, if you want a large root system, you need a large bacteria system.
You do.
Wow.
And on the root tip and all the pictures you're showing, there's always kind of like a little bulb where it's bigger. Is that because of after seeing that video, is that because of the accumulation and then it's with the ethylene, then it's causing it to then eventually stretch that part out? And there's always going to be a part that's it's bigger where the accumulation's happening, or like even in that picture, you can kind of see, there's a slight difference. It's not that part of the shadow, like it gets bigger. I
will tell you, you guys are very perceptive in seeing stuff. It's actually part of the mechanism. And what happens is these hair tips have thin walls, like the thin walls on the meristem, on the root tip meristem, right? So on these hair tips, there are thin walls, and it's like a plastic, like a elastic, right? And so what happens here, because these tips are thin, the bacteria, I guess I can show it better in this next image. Okay, so showing here, what happens, you can see this diagram, you can see this red is the cluster. This is where ethylene gets secreted. It causes an expansion in the growth. And when that grows, then it pushes out these microbes out of little breaks, little pores that form in this tip. Well, as it turns out, okay, so then you see the beginning here, these little blue dots now, and the C here, these are the microbes again. This is without all the microbes in the tip. Okay, so then it builds up these again. And what happens is, because this wall is thin and elastic, rather than these microbes, rather than being constantly rotated around, they will go out here in this little soft pocket at the tip, right, where the wall is like elastic, and they'll get stuck out there in that little pocket. And they'll just keep, as the plant builds them up, they'll just keep accumulating out there until there's a bunch. And then when there's a bunch there, they'll produce enough ethylene to then cause that growth spurt again. And you'll have that growth spurt, and they'll be squirted out again.
It's like an internal like pressure relief valve, and then the system kind of restarts.
Yeah, I was thinking the same exact thing. Exactly.
It has an accumulation, and then there's like a pressure difference, and it contracts, and the cycle starts again, or almost like that's how I'm visualizing it in my head right now.
It is, and we're very much, in a modern era. I'm just like you guys. It's a machine, a little machine, and we can view that, almost like mechanics of it, and looking at the mechanism of the system. Very much reductionist modern view of things. I mean, we don't see it as magic. Unfortunately, we don't see it as magic, but it's really a cool system that plants have developed. This actually is a tomato root hairs, and you can see the bacteria coming out. You can see how.
Wow! Look how it shuts it to the end. Wow! Yeah,
you can see it number 3. Look at number 3 there, and you can actually see those 3 little holes. It's like 3 little holes as it does it again.
It's like pooping
at the tip. It is like
Sorry.
Yeah. And this happens repeatedly, like that. Every time there's a growth spurt, right? You have this kind of ejection, and this is, this happens. There's other cellular processes that are involved here. For example, potassium is probably involved because these hairs have a vacuole in there, and the swelling is probably happening because the plant cell is putting in order to make the growth is putting potassium into that vacuole in the center of that hair. And that causes water to rush in. And then that causes this whole swelling to kind of go to the end as water rushes in and causes the bacteria to be ejected. So, I mean, it's all kind of the plant physiology, the kind of mechanisms of plant physiology that are involved. In this whole
so when we look at number 2, and as you can see, there's like a void as it's pushing it up. Is that water pressure, or that's that center void that you're?
Water pressure. That's water pressure. That because the bacteria are around the sides of the cell, right? So what's happening is the water pressure inside the plant cell causes the hair to expand, and that causes the bacteria around the sides, around the outsides of it, right, to be squeezed up. And as they're squeezed up, some of them come out the tip. You see how that works?
I love that. I love that. It's
yeah.
Wow.
It's crazy. You got to admit, it's crazy stuff. This is crazy stuff.
It is.
Yeah.
Especially how they use the water. You know, water goes up the tree, it goes out the roots, it goes in the leaves. That's amazing. I'm sorry. It's just little things like that.
It is. It is. I mean, the whole plants are fantastic living microbe processing machines. Okay, so what nutrients? Turns out most nutrients that we've examined are impacted by rhizophagy cycle. This is a study that a visiting scientist, Dr. Shuai Zhao, did in the lab using winter wheat. And he basically removed the bacteria from the wheat and then put them on one at a time and measured nutrients when he put them. Okay, so this is the control. This is wheat without the bacteria. You can do that by sterilizing seeds because they're seed transmitted. So he sterilized the seeds real well, germinated them, and you can see the roots are small, the shoots are small. Then he put one of the bacteria back on, a bacillus, common bacterium that people use in our as a biostimulant bacteria. And you see these everywhere, bacillus species. They are everywhere, really. But he put it on, and you can see the roots are bigger now, several times bigger. The shoots are bigger too, like 4 or 5 times bigger. You know, it makes a big difference in development. Here's the nitrogen measured in the leaves. You can see nitrogen with the bacteria here to the right, these 3 big bars, compared with no bacteria. And you see it's about a 30% increase in nitrogen. So I don't want to give you the idea that the only nutrients plants get are from these bacteria that they internalize in the rhizophagy cycle, because they're also getting nutrients from mycorrhizae. They're also getting them from solubilized nutrients in the water in the soil, right? If you squirt nitrogen in there, you know, they can get that. But it turns out if you do that, plants then won't get nitrogen from these microbes. They'll stop cultivating microbes. So you could ruin the system for plants if you put inorganic nitrogen.
That's interesting.
But are the plants getting the nutrients? They're getting the nutrients after the bacteria attaches an acid to them or something, or changes the form of the molecule of the mineral, the liquid. It's the water soluble mineral, right? It's not just a water soluble mineral. Then the plant take. Well, I guess it does. It's hydroponics.
So let me tell you how we think they're getting the nutrients. We think they're getting the nutrients directly from the microbes by removing the cell walls. And the cell walls, you have like nitrogen in the cell walls. You also have metals, iron, and other nutrients are vectored on cell to stick to the bacterial cell walls. So when the plant takes those cell walls off the bacteria, it's getting all those extra nutrients that are sticking on those cell walls, right?
Wow. So the minerals are on the cell wall of the bacteria, and when they're being stripped, they're being mined or extracted.
Some are there. Others come from the cytoplasm of the bacteria. Okay, for example, potassium. Okay, potassium is one of the solutes in bacterial cytoplasm. And so in stripping the walls and exposing those protoplasts to superoxide, it porates the membrane of the bacterium. And so more of the nutrients can leak out. from those pores. So that we think that's happening. And so you see, in some cases, you see a lot of potassium coming from rhizophagy cycle. Others, we see some phosphorus and nitrogen and zinc, and but we think that all nutrients are impacted. We can see changes in all nutrients. Phosphorus, here's a similar thing. Okay, you can see no bacteria that much, with bacteria that much. And of course, there's other ways that plants get phosphorus. I mean, some of these microbes are phosphorus solubilizing microbes. So they will go out and solubilize phosphorus. It'll go into the water, and then the plants can just absorb that, right? So plants are doing everything to get nutrients. This is one of the things that they're doing. Potassium, here's what I said, potassium is potentially important. Here's one of the bacteria, this L1, Bacillus species 1, and you can see this big bar for all that potassium it gets from that particular bacterium compared to these other Bacillus species, which get less potassium. Any nutrient we look at, we can find some differences. We can't exactly say precisely how for any given nutrient the plant's getting it using microbes, but it's connected with the presence of the microbes.
So can I say, or am I safe to say in my mind, after the no bacteria, introducing individual bacteria in most applications, like, are we over foliar spraying like our farm fields and stuff like that? Like if we don't have the bacteria and then we're just foliar spraying all the time, nitrogen and stuff like that, then we're not fixing, like you show, no bacteria still has an intake, but then when we introduce these at different levels, we increase the availability, but well, that availability might become less and less throughout the years if we keep doing foliar sprays, in a sense. Like that's kind of how my mind's thinking, like where we're not conditioning and fixing the soil, we're just fixing the in-moment problem.
We are, I mean, it's a good point, Dave. We are negating these systems in plants completely. When we put nitrogen, for example, we turn off plant exudation, and they'll no longer attract bacteria to the roots and internalize bacteria. You, in a sense, you then make plants dependent on the nitrogen because they no longer can get bacteria into the roots. And any bacteria that are in the soil, if you put nitrogen on the soil, it will stop nitrogen fixation because the bacteria will just get that nitrogen too, right? So, and these microbes, I'm about to tell you about these microbes that go into leaves, and putting them on the foliar applications now that you're talking about, negate or paralyze these systems in leaves as well. So application of any type of inorganic nitrogen to plants, and and I know what all those forms can be, I mean, but mostly we're talking about ammonia, we're talking about nitrate, for example. Putting those on leaves or on plants will make them more dependent on application of that form of nitrogen. And that has side effects too, because what happens when these plants are getting nutrients from microbes, because they're using oxidative interactions with the microbes, oxidizing them with superoxide, then plants become oxidatively resistant. They get antioxidants and phenolics and chemicals that make them tough, hardy, right? So they're oxidatively stress tolerant, they're resistant to climate change, they're resistant to heat, excess heat or cold or whatever, they're more resistant. And if that's so long as they have their microbes that they're interacting with. But if you negate that, give them just ammonia or nitrate fertilizer, then they don't use that oxidative interaction. They just suck that up out of the environment, and then they become weaker. They're not as hardy. They also, by the way, won't produce as many secondary metabolites. For example, if you're trying to grow cannabis and you want to maximize your cannabinoids, you don't put inorganic fertilizer, you don't put nitrates on them. You instead cultivate them let their microbes internally function and make the nitrogen for them, and then you'll have more cannabinoids in those plants. Okay, so it affects the hardiness of the plant. Okay, if they're not oxidatively resistant with their microbes, then they also will be more susceptible to disease, and then you have to because you're using this inorganic fertilizer, you're going to have more disease, and then you got to put more chemicals on. You got to put your fungicides there to control them, right? So it's a slippery slope down. Yeah. Down. I mean, that's where our modern agriculture has gotten us. I mean, I'm a scientist like everyone else. I mean, we're pushing plants to feed the masses, right? I mean, we don't know if we can stop pushing plants with fertilizer. We may be totally addicted to it so much that we can't stop. I mean, I don't know what it's going to be like, but people certainly with regenerative agriculture, they are saying, no, we can grow crops, get good yields, without using these chemical fertilizers. So,
well, we were talking about this a little bit earlier in the very beginning of this conversation was in the 70s, my parents, when they came into Monterey County, they the same owners of this land couldn't get, for the last 50, 70 years prior to this, they couldn't get past 1,400 pounds an hour of harvesting, packaging, and distributing lettuce. And why? What are some of those problems? Harvesting it, going to mold, I can't sell it to the end consumer. But we're talking about that, right? It's like, what are all these problems of harvesting, packaging, getting it to the end consumer? And there's certain areas in the world that there's 3 Mediterranean climates that produce roughly 6 to 7% of the food for each location. Just these 3 locations roughly produce 20% of the food for the rest of the world, right? So, and they're in these mecca climates where it's this perfect environment, but as you go a little farther out on its radius or its diameter, it gets a little warmer, a little drier. And so you have these mecca areas where all this food can be grown and distributed from one central area worldwide, right? And so, like you were saying, being able to grow like we're talking about chlorine and killing the microbes, because the microbes, just like you're saying, you take the plant out, these microbes stop moving. So there's obviously there's things that when something stops happening, other things get activated. And can food be like you say, can food be produced, packaged, and distributed to the end consumer with microbiology techniques, or do we have to shift our distribution techniques? So no longer trying to grow so much food in a local area, but doing a better job at distributing our local food to our local community if we're going to implement these types of techniques. I don't know, but I'm just...
Yeah, no, that's a good question. Those are good questions. I mean, that is the relevant question. I don't have the answers for that. My answers come exclusively about what these microbes are doing in the plants and how common they are in plants and the mechanisms and so forth. But I mean, the questions that you ask are really the relevant questions for, that are necessary to resolve to possibly get us out of this dependence on agrochemicals, right? That can we use these microbes? I will say that just because people are familiar with this, what you see in plants is very much what you see in humans when you kill these microbes. For example, the dysbiosis that people get when they take too much antibiotics, and they take a bunch of antibiotics, and then they'll end up with intestinal bowel disorders or Crohn's disease or something like that from doing these antibiotics. That dysbiosis happens because what happens is the antibiotic kill out. I mean, everybody knows this already. Kill out the good bacteria in our guts, right, and leave only the bad ones, the Clostridium difficile, the endospore formers that are more resistant to the antibiotic because they form endospores and nothing can kill them when they do that, right? And then the good ones are all gone, the bad ones are there, and then we're sick, right? Well, the same thing happens with plants. Okay, one of the worst things you can do to a plant would be to treat the leaves in such a way that you kill all the microbes in association with those leaves or in whatever crop it is, because then you get rid of all the beneficial microbes that are on it, and you leave only, other fungi, other organisms that can come in. And one of them is, for example, Fusarium, a fungal pathogen that'll come in and then cause rot in your plant. So you develop a dysbiosis by trying to sterilize them, which has to do with your example.
Or just like what you and Dave were talking about, making the foliars too strong, it will make the microbes dormant. So in a sense, maybe not killing them, but make them dormant and not having those same benefits.
Yeah, that's right. That's right. Negatively impact them. Yeah.
Mm-hmm.
So, okay, so I want to turn my attention now, because I said that plants are these microbe processing organisms, machines, microprocessing machines we discussed. And they actually take the bacteria up into their leaves and use them in their leaves to fix nitrogen. So they're getting other kind of nutrients now from these microbes inside their leaves, and that's what we'll talk about. And one example here is the nitrogen fixing endophytes in hosta. Is that the one? Oh, I think in general the same chemical interactions happen in roots of plants that we're going to be talking about in the leaves now. And the chemical interactions, I didn't emphasize them before, but you have your ethylene production, you have your superoxide production, right? And the interaction, so the superoxide is the plant product, plant cell, and the ethylene is the microbe cell. But you have another reaction here that happens. And this is the microbe will produce nitrogen, and we think it's nitric oxide. Could be some ammonia, but we think it's nitric oxide. It will produce nitric oxide. Nitric oxide is an antioxidant. So the microbe will produce nitric oxide in order to defend itself against superoxide produced by the plant, this 2O minus here, superoxide. And then that will end up, those two will combine and you'll get nitrate. And nitrate is the fertilizer, right? So this is how plants, in leaves of plants, and also in some roots, you have these interactions happening so that the plant then can get these microbes to release nitrogen, and then it will have a form of nitrogen into the leaves directly. Go ahead.
Is the plant producing the nitric oxide, or is it getting the nitric oxide from the electroconductivity of the atmosphere, combining to the bacteria and then transferring it to the roots?
So what happens is,
does that make sense? Was that a?
No, I know where you're coming from, because you hear a lot about nitric oxide and where it comes from, right? So it's a little bit confusing. So plants produce nitric oxide in their tissues, and they produce it as an antioxidant and also as a growth hormone. So it's a signal molecule for plants, but it's also an antioxidant. So people also have this nitric oxide, and in people, it is an antioxidant as far as I know. And you have it, for example, if people have, in heart disease, sometimes in vascular disease, sometimes you have nitric oxide as one of the players, but nitric oxide tends to be an antioxidant. So it's in these systems, like plant systems and animal systems, right? So it's naturally there. It's naturally produced in those organisms. Well, in the respect of endosymbiosis in plants, it's produced by bacteria. And the bacteria produce nitric oxide in response to the plant producing superoxide. And so the bacteria, nitric oxide acts as an antioxidant, and it will combine with superoxide, and it will form a combined chemical, which is nitrate, which is not oxidative and not damaging to the plant. So you have plants using nitric oxide to protect it, or bacteria using nitric oxide to protect itself from the superoxide produced by the plant. Does that make sense?
Yeah, that's crazy.
Wow.
That's cool. It's like that one's really good.
Amazing.
And then these combine and you have nitrate, and that nitrate is the fertilizer that the plants then absorb. So this is naturally how plants are getting nitrogen, right, from microbes. Well, we humans discovered that we can, instead of doing this interaction inside with microbes, because we didn't know that was happening, that we can make nitrate, and then or ammonia that can then be formed into nitrate in this by soil microbes. that we could use nitrate and give plants nitrate directly so that they don't have to do this microbial plant cell interaction, right?
But if you do that, you're jumping the gun, you're skipping all the steps, then you have to go back to the beginning. Then the nitric oxide is affected, less produced. Then it's not. Correct. Then the bacteria is not being protected as much, so that it's like
the plant's
so,
not being protected as much either.
Right. So then you can kind of create an overwhelmed system where those checks and balances are, when they're gone, it's now what happens when that's gone? What are the superoxides doing? Just like cannibalizing itself at that point, because it doesn't have those oxidation processes to protect it.
Well, and what happens is, because if you just give them nitrate, they don't do this, right? They don't make superoxide. The microbes don't make nitric oxide. They don't make antioxidant. Plants aren't making antioxidants because they're not doing this. They're not making any phenolics or any chemicals that'll protect them from oxidative stress, right? And so they become stressed out plants. You give them nitrogen, they get lazy. They don't do this.
They look green, though. They look green.
They look green, but they're weaker.
In addition to this, tell me if I'm wrong, but all these chemicals that are providing nitrates, it's not just NO3 negative. They're providing there's extra carbons in there. There's extra a whole bunch of other crap in there to provide the nitrate. And what's happened to all those excess charges, right?
It's all of this that's happening in there, right? All of this is not happening if you give it nitrate. You don't need to do this. They're not doing this. You had carbon dioxide forming, you had hydrogen peroxide forming, ethylene that causes root growth. You're not stimulating root hair formation. You're probably not growing trichomes as well. Plants, you give them nitrogen, but they're weaker. You remember there was, I don't know if you remember, there's some work that has been, you hear about it in the past few years, where they're doing these nitrogen, carbon dioxide experiments where they're putting carbon dioxide into plants and showing that as carbon dioxide increases, plants grow faster, but they're having less nutrients. Well, what's happening there, we think what's happening there is that carbon dioxide is actually inhibiting formation of superoxide, inhibiting that superoxide formation. And so you don't get plants getting as many nutrients out of microbes because they're not making as much superoxide. That reduces.
We were just talking about that, Mark. Remember the other day absolutely. I was bringing that up, that we were watching those videos with John Kemp's and how they were doing those CO2 applications, and it was killing off the microbial lives. And then they went in the lab and they isolated seedlings and just barely increased the levels, and the bacteria was like died off. John Kemp, sir, right?
And
it's really interesting because when you kind of just look at it, obviously one's acid, one's alkaline, so it's neutralizing itself, right?
Yeah.
When you have too much CO2, it just neutralizes the chemical reaction, so it makes everything inert. That's how I see it. Am I wrong when I see it like that?
I mean, I think you're right in the sense that you should look at this as in one respect, you should look at it as these simple kind of chemical interactions that happen. But in the case of the effect of carbon dioxide on superoxide, you're really talking about the effect of carbon dioxide on that. Remember the enzyme I showed, NADPH oxidase, right? Okay, if there's not enough oxygen around or there's too much carbon dioxide there, it competitively inhibits the formation of that superoxide, right? High levels of carbon dioxide will inhibit that oxygen, the superoxide formation from that particular enzyme. So there it's a specific enzyme. At least I think that's how that's what we think is happening with elevated carbon dioxide experiments that are actually reducing nutrients like zinc and iron being acquired into plants, and even nitrogen. So it's this inhibiting this process basically is what's happening, and that's because plants evolved with a certain level of oxygen and carbon dioxide in the atmosphere. If it gets too high, that's going to screw up the systems that they use. Okay, so anyways, that's how we think about it. But it is, you're right, there's this other soil chemistry that's going on that could play into this as well. And one would just have to, think about how that, study it a little bit, analyze it a little bit closer to see how that figures into it. So what we did in these studies is to look at leaves and look for these symbiosis in leaves. And we looked at like 30 different species of seed plants, of monocots and dicots, grasses and agaves, and hosta is one of them. And cannabis is one of them. Hops is another one. Tree of heaven is another one. And we look for those chemical interactions inside leaf cells. And we found them inside cells that didn't have photosynthesis in them. So some cells, like for example, the epidermis of plants doesn't have any chloroplasts there. And trichomes don't have chloroplasts in them. And so we think that these trichomes evolved in order to cultivate these microbes internally and get nitrogen from them. And I'll explain that a little more. And because what happens is, and the reason we don't see these in cells, leaf cells that have chloroplasts, is because oxygen, chloroplast means photosynthesis is happening. And what happens with photosynthesis is that water is being separated and oxygen is being produced, right? Hydrolysis is called. That happens when water is broken apart in photosynthesis to get electrons. And then oxygen forms. It turns out that the oxygen that comes out of photosynthesis inhibits nitrogen fixation by bacteria. So they're two incompatible processes. Photosynthesis and nitrogen fixation are incompatible. So they can't be in the same cell types. They have to be in different cell types. So, but plants, in plant leaves and flowers, they produce both cell types. And we'll look at those. And so you see what I'm talking about.
Does nitrogen's fixation happen more during the evening than during the day?
So there is some, it's beautiful.
Is that a credible question?
Oh, of course. Oh, we've been trying to figure this out for a while. There is data from the 80s by another Rutgers scientist, a guy who died many years ago now. When I first came here, he died, a guy named Carlos Neira. But he was looking at corn. And what they found is that during the daylight hours, you can find nitrogen fixation occurring in corn. At night, it stops. So he suggested, and I think some others have similarly suggested, there is this diurnal effect. And quite possibly why we see that diurnal effect in some plants at least is because during the daytime, you need sugar. I mean, you're doing photosynthesis and you're producing sugar, and nitrogen fixation also requires a lot of sugar. And so in the daylight hours, you get that sugar. And as it turns out, that nitrogen fixation happens in cells where oxygen doesn't accumulate. So you don't have to worry about too much oxygen in those cells at the time, right? Because they're being excluded physically from those cells where the bacteria are located and nitrogen fixation is happening. I mean, what you just have to remember here is that oxygen inhibits nitrogen fixation. That's what you just have to remember. And so
it just sounds like some of the chemists are going to sleep, I'm just kidding. I'm just joking. They're just going home, not doing work.
Yeah.
Be back tomorrow.
See you tomorrow.
Oh,
yeah.
Yeah. Well, so this is hosta. Hosta is a cool plant. You don't put any fertilizer on hosta, at least you don't need to. Part of the reason why is because this is the epidermis cell of hosta. And you can see right in the middle of that epidermal cell, you can see the nucleus. See that big thing I'm circling around? That's a nucleus. Well, it turns out the nucleus is full of bacteria. And it puts sugars into that nucleus. This is the epidermis right on the surface. There's no chloroplast there. So it's low oxygen. Okay, it puts sugar where those bacteria are. Then little by little, it will release those bacteria out into the cytoplasm from that nucleus, and that's where it starts extracting nitrogen from those bacteria.
So it's just a different evolutionary method of farming that this plant created for itself.
Correct. In the leaves, these are hosta leaves. And you can see it over here to the right too. You can see it, this epidermal cell, and you can see
that's why they're so resilient, probably then, right? And they don't need much nutrients. These are plants that you tell people that they say, I don't have a green thumb. I can burn water. And you're like, here, try a hosta, try a spider plant.
Yeah, that's cool. Yeah,
right.
They don't need stuff. In fact, if you gave them stuff, you'd probably mess them up.
Right.
Mess them up.
Are these the types of plants you normally find in a completely enclosed aquarium? What do they call them? Aquariums, where you put
terrarium.
A cap? Terrariums, yeah.
Terrarium. Hosta, these are the kind that you find that gardeners put around the houses. So everywhere. Along the walks, they're everywhere. Hosta,
like cichlids.
Yeah, fish.
These are like, you don't have to be thoughtful. You can take a shovel, dig some out of the ground, put them in the middle of your yard, and a week later, it's just like, it grew. Exactly.
So this is one of the symbioses that you see. That's what we call the nuclear. Now, okay, I'm going to say, instead of symbiosis, nuclear symbiosis, we could say nuclear endosymbiosis here, right? And because this goes inside the cells. Okay, this actually is a movie of a hosta, and you can see the nucleus there, and you can see now this is stained for superoxide, and see what happens. The blue, see the bacteria coming out of the nucleus. Can you see that? Little bit at a time, and they're staining blue, and that's because as soon as they come out, they get hit with superoxide. If we stain for ethylene, we'd see they're secreting ethylene when they come out too. And so you can see little by little they're coming out. And so what happens is inside the nucleus, the plant gives them sugars. They also have a membrane around here that excludes some oxygen, right? So it's low oxygen. And they give them sugars. They can fix nitrogen. They can replicate. And then when they come out, then that nitrogen is harvested from them. And they're little by little. And then what happens, they come out and then they're circulated. You can see the cyclosis happening here. They're circulated around those cells. And eventually they're fully degraded too. They're fully degraded. So these plants are, growing these microbes in this special cell layer called the epidermis, and it turns out hosta also has these variegations, these yellow areas or light green areas where it doesn't have chloroplasts. And so in these variegated areas, hosta, in the leaves of variegated areas, or the variegated areas of leaves, the yellow areas, it will also have these nuclear symbiosis so that all of those cells then can get nitrogen. So, and some work with pelargonium has actually shown that plants that are variegated are more nitrogen efficient than plants that are not. They could survive without nitrogen more than plants that are not variegated. Okay, and it's also been shown that if you take nitrogen, this talks about the feedback mechanism, you feed them these variegated plants, some species, I guess it's pelargonium, I can't remember which one now, but give them inorganic nitrogen and the variegations will go away. So plants may be variegated plants, may be having these variegated areas specifically to get nitrogen out of these microbes. And because they can't, they need low oxygen. And when you have chlorophylls there, that inhibits nitrogen fixation.
This is like the coolest video I've ever seen in my life.
Yeah,
isn't it?
It's so, dude,
it's screw Hollywood.
There was one point, it almost looked like one of the bacteria is like dipped down, but then all of a sudden it popped back up in the other part of the cellular structure. I was just like, wait, where did it go? Oh, there it is. Like, wow.
They're moving. These movies help us understand what's going on. Because, it's not a thing where you can just take a picture and you got to see what's happening, and then it sits into the brain. We have to see that.
How did you get the video on the roots that were traveling before realizing, like, you obviously probably removed the plant and looked at it like nothing's moving? How did you realize, hey, maybe we should look at it before we take it out of the plant?
So what we did was we grew them in agarose, right? We grew them in agarose, and then we turned the plant, the plate all upside down, and then we looked at it from the reverse of the plate. Yeah.
Got it. See-through glass,
And-
basically.
Yeah.
They grew in
and,
this. Okay.
and soon as I told you I was a mycologist, right? So
Yeah.
we use plastic Petri dishes, right? So for fungi. So we use them now for plants.
Oh, that's
We
cool.
use them now for plants. Okay, so grasses, the way grasses grow, we think they grow the way they grow. Tillers growing everywhere, roots kind of wild because they're roots, a lot of rhizophagy cycle in the roots, but also shoots that keep growing, lots of leaves everywhere. We think that's because in the growing cells of grasses, the shoots of grasses, they have nitrogen fixing microbes. And we have couple of images. Okay, for example, if you consider the epidermis of grass, they have these weird convoluted walls. And what we think this is actually an adaptation in grasses in order to get more bacteria that they cluster in each of these little bumps of those cells, inside those cells. And that's where nitrogen gets fixed. And I'll show you. Okay, so look to the right here. You can see those cells. Now you see the little like serrations there on those epidermal cells. These are young, but you see the things in between. You see the big black arrow there. Mm-hmm. Those are bacteria. Those are the bacteria that, and you can see, look at this one here. You can see the two rods in there too. You can see that. Those are bacteria. And that one happens to be stained for nitro blue tetrazolium for superoxide. So they're being hit with superoxide right there.
Wow.
These are the we're going back to those older slides. These are the conditioning areas where the superoxides are coming in. That was the side diagram that you had before. This is the actual view of it now.
This is looking down on the grass epidermis, right?
And they're being stripped now or later, or they were already stripped? So they're being stripped now, right?
So we don't see the bacteria in this picture. Just grabbed this off the internet, right? But if we look at the seedlings that we looked at, this is a red fescue here. And now you can see the same cell type. This is on the epidermis, and you can see the bacteria in those little
I was referring to the one that was like a computer animate, like where you said you were put your artistic part into it, and we're talking
way
about
up. Yeah.
So like that's an actual view of that, right? Like
yes.
Where it's being held right there. This is where the superoxidase is. Okay.
Yeah, it's like that. It's like that.
Right.
So these bacteria in the grass epidermis probably come from the nucleus, and then they're pushed out of the cell, out of that membrane in the periplasm, right? That's probably what we're seeing here. It's hard to know for sure whether it's outside the membrane or inside the membrane. I think it's probably outside the membrane, like you. I looked at this and I thought, yeah, I think that's probably outside the membrane in this case. So it transports them from internal to the cell to outside in the case of the epidermal. And of course, this is important because you have these serrations like this. You can put your bacteria in those serrations rather than them accumulating at the end of the cell in a bunch where you can't get nitrogen out of them, right? So if you have them organized along the cell wall, you can get more nitrogen because you have them organized. And you just see the efficiency of it. The plant machine, this is the grass, the invention of grasses for getting nitrogen out of these microbes, the serrate epidermal cells. So this actually shows a mesophyll cell in a grass leaf sheath, and you can see the bacteria in this nucleus. Okay, so that this is stained for using copper sulfate, and we can see sugars with copper sulfate that'll stain orange, and you can see the orange in there with copper sulfate staining. That orange is due to the sugar around those bacteria in that nucleus. Here's another tree that this is a dicot now that has trichomes that fix nitrogen, and this is the tree of heaven, that tree that's everywhere, the one that you don't fertilize, it grows, in the pavement in the city streets and in the little,
it grows everywhere.
Everywhere. Tree of heaven now, it with no fertilizer or anything. Well, if you look at the leaves, here are the leaves, and you look closely at it, you see they're covered with these trichomes. And if you look closely at the trichomes, you see this. Trichome to the left, you can see they're filled, all these little bacteria in it. They're filled with bacteria, and you can see they make little pits on the side. This turns out to be a common way that dicots make trichomes. They have their little lateral pitted, and you can see the bacteria here coming out of the pits in the trichome. So these are growing, and they're fixing nitrogen in the trichomes. And out of older ones, they have these pits. I don't know what these pits do, but bacteria can come out of the pits. And the way that this happens, this actually shows the cyclosis again. Here, this is the development of one of those trichomes, and you can see the cyclosis. You see the bacteria moving around. So the plant is using cyclosis to move the bacteria into positions, into little pits on the membrane in the hair. And you can see them accumulating right in there in the pit. You can see these other areas where the pits are. But then it extracts nutrients from the microbes in those pits, in these clusters. And the holes in the wall form just over these clusters of bacteria. So that, and that's where some of the bacteria will come out. We don't know the significance of bacteria escaping, but this is actually a very efficient.
Did you say bacteria were in the trichomes of our plants?
Yes.
So they're in all trichomes.
Most trichomes have bacteria in them. We see some trichomes that form that don't have bacteria, but most trichomes appear to contain bacteria. Like here's another one.
What plants did not have bacteria? Do you know?
There was a little mint that has these long, it's called a mule ear, I think, that has these very long, sinuous trichomes where we didn't see any bacteria in it. But the mints have glandular trichomes that are loaded with bacteria. So they have different kinds of trichomes, and some types may not have bacteria. Other types definitely do have bacteria.
That's interesting. So would you say that the ones that don't have the trichomes are those trichomes, or are they a different part of the plant?
Well, we name them all. We call them all trichomes. A trichome is this class of cells that people have just considered plant hairs, and people have theories about how plant hairs work. In some cases they're thought to be like little anti-insect things where maybe insects won't eat through them, right? Or maybe they'll produce some chemicals that will deter insect feeding or something. So we don't really know. We have all these ideas about how they
It almost seems like a big part in their immune system, like an external immune, like the upfront part before it actually something can come inside it and affect it. Ultimately is how I'm kind of viewing the hairs, and there's bacteria in there.
Well, all of those ideas are valid.
Yeah.
And we don't know. So your ideas about immune system are
like an external
valid
immune system.
And as timely, valid and timely, right? I mean, I think people are going to be asking more and more questions about the functions of hairs and how plants use them and what they're really for. We're going to look more closely at these trichomes on plants, I think.
Could I share a holistic thought process to this? I've been sharing for years.
Yeah.
It's like have you ever heard of grounding, where you take your shoes off and you go into the soil? Have you ever seen the blood analysis, what it looks like before and after?
No, not blood analysis.
So,
I mean people blood flow into the feet and.
Well, no, what they find is a negative charge begins to happen around the blood cells, and they begin to freely flow. Grounding. So if you to look at the underneath a light blood microscope, you'll see that there's coagulated blood, or it's just kind of stuck together. And then after they ground, there's a negative ionic charge that the blood cells begin to flow. So over the years, I began to think, there is like this natural communication that Mother Earth or the microbes are providing to its organisms, right? And how is this connection happening? As we talk about these bacteria connecting to the cells and traveling on the outside of the cells. And I think like how bacteria of plants and us, they say like we're like 90% microbes and 10% human or something like that. So it's like constantly thinking that communication between what does Gaia or Mother Earth want us to do, right? And we're the little microorganisms living on the surface, and we're just disconnected from her. We're not listening to her in a sense, the ether, the greater knowledge out there, because we cannot pick it up. But then I think about these root hairs as well, or like the signaling of the hairs on a cat, right? There is a sense, is a sixth sense or additional sense. And I've always thought about it. It's like the plant is, connecting with the soil, right? And even as it's further extending out into the atmosphere, it's extending its whiskers or its hairs out to further connect to the earth or to the ether or whatever. That's my holistic thought process to it. But please continue on that.
Well, I think I agree with that idea. In that, I mean, especially the last part that you said, that relates to these hairs and the way the root hairs work, that extending into the soil to interact with the soil microbes and with the soil particles, but also extending into the air now, leaves and leaf hairs, right? Like root hairs, that they are functional in absorbing nitrogen, for example, and from the atmosphere, and then putting that in the form that the plants can then get as nitrate from those microbes. And then that's transmitted to the rest of the plant. So it is, and it is like, I mean, you had, it was, well, I mean, you also mentioned something else. You also mentioned something regarding the electrical charges that go into the human body when we walk on the earth, right? And that affects the physiology of the organism, right? Well, we don't know anything about the electrical charges that are impacting these microbes in the plant tissues, right? And in fact, it may be another idea, may be that with, for example, microbes going into the root cells, that they may be partially attracting those microbes using electrostatic attractions, right? Because the microbes in the plants are different charged. And if you look at the meristem tip of a root, for example, or the meristem tip of a shoot, those are going to have charges in them. And so the plant could be using those charges to put those microbes in the right spot, take them in, like that. So the same charges could be affected. And with the Gaia idea, yeah, the Earth is a giant organism, and we're not connected. We're not connected to functioning to the nature of how that organism functions, and we're just now waking up to it. And the way that plants actually acquire nutrients using microbes possibly can be another bit of information that helps us to change agriculture and stop, get a little better connection, right, to Gaia, right, to the earth organism. See how that, so we take care of it a little bit better. So that's the and I fed off of what you said, Mark.
Thank you. Thank you. Yeah, you're not making me look a little crazy.
You're welcome. No. That's, you're not. Actually, there's interesting stuff on that Gaia channel. So I've seen that too. It's interesting stuff, even about the barefooting, right? Earthing.
Earthing. Yeah, grounding. Yeah, absolutely.
Yeah. No, it makes me want to do that now if it weren't so cold. So, anyways, one of the plants where we see these glandular trichomes that have, it turns out that we think this is an advanced type of endosymbiosis, glandular trichome, because it has the chemistry involved. But here's a diagram of a glandular trichome. It has these monoterpenes in it, right? These cannabinoids that develop. Also, hops has these terpenoids in it. A lot of dicots will have terpenoids in their glandular trichomes. Well, they also have lots of bacteria. Well, this shows for cannabis. Actually, you can see they have a stalk, and that stalk is green. You notice that green color. That's where photosynthesis is, in that green part. And you see the head, you see it's not green. Okay, that contains the microbes and also contains the plant cells. That's the reactive chamber in that glandular trichome. And so what happens, the way this works is these stalks will make the photosynthesis that gives the sugar that goes into the heads.
This is what's happening in the root system. This is what's exactly happening in the root system. It's getting to the end and it's swelling up. Exactly what you showed us earlier.
Except that this is, yes, it is very similar to what's happening in the root system, the chemical interactions, but it's not ejecting these microbes out of these trichomes. They stay in there. You'll see it is different, but yes, it is like what's happening in the root system. There are parallels. In fact, we understood the chemical interactions that are happening between plants and microbes. We understood it first from the roots, and we described all the chemistry there, and then we applied it to the leaf trichomes. And I'll show you that. Okay, so this is our cannabis glandular trichome. You can see the little green that's underneath. You're seeing between that, through that head, and it's underneath the green. That's the chloroplast in the stalk cell, right? And you see these red arrows. These are where the bacteria are. This is a developing glandular trichome in hemp, and it's stained for superoxide. So these are little clusters of bacteria. I'll show you a close-up of it. Now you can see the bacteria in there. They're rods. Can you see the little rods in there? And you see the purple around it, and that's the superoxide. So the plant is putting superoxide on these microbes, and that superoxide is going to start extracting nutrients from the microbes, but it's also going to make these microbes into protoplasts that it can then replicate and then continue to extract nutrients from. So it's going to make a lot of copies of these. Let me see if I can go to the next one. This is what a glandular trichome looks like of cannabis once it's more fully developed. And you can see the cannabis trichome cells here in the middle there. And you can see the green underneath, that's in the stalk underneath. So this is pressing down on the top, looking at the top. And you can see the blue there, that's where the superoxide is. And these are the microbes around it. So all this thick stuff is microbes in there. And what will happen as time goes on is this glandular trichome will accumulate cannabinoids that'll get in there. And we think the cannabinoids actually are antioxidants. They are going to protect these microbes from oxygen in the atmosphere. They're going to absorb it. Now, that seems inconsistent with what we're seeing here. You see superoxide. But remember I said superoxide is very short-lived, very potent, but very short-lived. So that superoxide isn't going past this little layer. So you don't see it, say, out here, right? Just right in this zone right here. Okay, so that's the local zone where the microbes are being hit with superoxide. But out here, they're not.
It's so. Oh, sorry, Dave. Go ahead. I'll just get it out real quick. I'm sorry. No, go ahead, Dave. Sorry.
No, go ahead.
It just sounds so amazing because it looks like the microbes can change the pH internally within itself in just like centimeters and millimeters of a space to do a chemical reaction, and then have a completely different pH happening just right there, in the same cell. Dave, please go ahead.
Let him answer.
This is fantastic. It's like on a nano scale, right? This is a little nano machine, a little nano nitrogen fixing machine is what we're saying, right? This is glandular trichome. Now I will show, this is the model for it. And so you see the red is the bacteria, and the nitrogen flow, you can see the blue arrow going from bacteria to the plant cell, right? And the carbon flow would be sugars coming from the plant cell going to the microbe to replicate those microbes and to fuel nitrogen fixation, right? Nitrogenase needs sugars in order to get nitrogen from the air and put it into nitrate or ammonia in forms that can be used by the plant. Okay, and then we have the third element in these glandular trichomes, which are these chemicals. And people have thought that these chemicals are largely maybe defensive property. They have defensive properties. Maybe they will keep, like for example, in Canada, maybe they will mess up the insect will eat it and they'll get kind of confused mind, and it won't kill them, but it'll keep them confused. The problem is animals tend to like eating this, so they would keep coming back. They keep coming back. So that's a problem with that idea. Okay, but it's also known that many of these terpenoids and alkaloids and flavonoids and phenolics and other compounds that are inside these trichomes also are antioxidant. And so what we think is that the role of these compounds may be to scavenge oxygen, little free oxygen, and that will reduce the oxygen around these bacteria and enable them to fix nitrogen more efficiently. So it reduces the oxygen inhibition of nitrogenase.
Can I
Dave,
ask before I forget? So
go ahead. Yeah.
Okay. It's blowing my mind now that I understand that there's superoxide, that whole bacteria process is happening. So what I want to lead into is what causes eventually the trichome to turn, at least I'm going to speak for cannabis, for that amber color. So is it that superoxide process is slowing down, and then now in my mind I'm thinking there's more of the nitrogen coming in, or that process degrading it, causing that amber? Or
are the bacteria producing keratines?
So the chemicals, keratines could be in there, by the way. That's another flavonoid that's an antioxidant compound. Okay, so but these chemicals are typically yellow color or orangish color. They could be colored, and so I think that's what we're seeing with the. Let's see if oftentimes, I don't know if I have the right picture, but I don't think I have it.
So you're saying that an accumulation is what changes it through density?
Yes, accumulation of the chemicals, the terpenoids and the other cannabinoids and the other alkaloids and flavonoids that are in there. Flavonoids typically they're typically yellowish kind of color. Yeah. I mean, you really have a mix of antioxidants, and that's what you have inside these glandular trichomes. In fact, people have looked at the genomics of transcriptomics of trichome formation, I think, in I don't know what plant, tomato maybe. And they suggested, because of all the antioxidants that are accumulating there, they suggested that there's a lot of oxidative interactions happening, reactions happening inside trichomes. So you can look at the literature and you can see allusions, reports. No one, like the bacteria are missing from the equation because no one sees they're there, but you can see where their research is pointing, there's something happening oxidatively in these trichomes. And, if you put nitrogen on these plants, inorganic nitrogen, then you no longer get all those chemicals formed. So there's evidence, kind of surrounding this, but no one sees the microbe right in the middle.
Wow. Yeah,
no, it's really interesting. It's really interesting. I mean, it tells you something about people. We are blind. And I said this is the second time at least, we're blind until we first see something. We're blind to it. We have no clue. We don't understand. We have every other explanation until we see it the first time. Right. And then suddenly you can see it everywhere.
Ignorance is bliss.
It is. Here's my student again, April Meachie, working on hops. It's easier to do hops. These are the cones on hops. Turns out these are covered with glandular trichomes. And so that these hops plants are producing nitrogen actually right when they make their fruits. They call these cones, right? They're full of these same kind of terpenoids that we use in beer that you see in Cannabis. So comparable. This actually shows one of these glandular trichomes. The red arrows there are showing clusters of bacteria. And this one, I guess, stands for ethylene. So it shows the bacteria are producing ethylene in there. Here's a close-up of that. This is nitro blue tetra. This is superoxide now. You can see the bacteria here. That's the superoxide around those bacteria. This is early development. Hops. Here's a hops trichome, glandular trichome on the root of on the little bract actually in that fruit, in that hops cone. And you can see the regular cells. You can see this is what they look like. They're not very dark, but then you see the trichome. See how dark that is. This is actually stained for nitrogen, using a nitrate stain that makes it blue when there's nitrate. So these glandular trichomes really get filled with nitrogen. And we oftentimes the chemicals that are produced in these, like in Canada or in hops, or another one is citronella. Citronella, I don't think I have a picture. I should get that picture to show you guys. I can't show it. I don't think I have it in here, but the chemical will just cover the bacteria so that the bacteria, it's almost like a lens. You see a layer of chemical, of terpenoids, and then you see a layer of bacteria, flat layer, and then you see the plant trichome cell underneath that. So the bacteria are sandwiched between the plant cell and the layer of chemicals. And so that layer becomes like a shield, an oxygen shield on the, so it's, anyways,
three sites back, you had that one right there. Hobbs stained with ammonium malignate.
Yeah.
Malignate. I always butcher that
ethylene,
word too.
That's ethylene stain. That's ethylene.
So the ammonium malignate to show, so the ammonium malignate is ethylene?
It will stain ethylene blue. So it's a stain that we use to detect ethylene.
Because ammonium malignate is a chemical that I have, so there's four malignates. I'm sorry. Okay, guys. Sorry for the whole channel. I hate this word. There's sodium malignate, there's ammonium malignate, there is one other, there's like four of them that they put into fertilizer bottles right now to help provide a malignate to the plants, right, as a trace mineral. And molybdenum. And the ammonium molybdenum is the one I've always encouraged to be, if you're not going to get your molybdenum from microbes, then they try to give it through these other forms. And I recommend to use this form versus sodium, because sodium builds up in the soil. I guess my question to this is the ammonium malignate, is that what the bacteria is produced naturally, or is this just something used as a stain to help, and it has no reference to?
It's the latter, Mark. It's the latter. This is just used as a stain. It's a way we actually devised this as a micro histochemical stain. It's used as a detector by agriculturists when they want to measure when their plants have too much nitrate. So they'll put it in there to kind of detect over nitrate accumulation in plant tissue. So we developed it for microscopy use. It's just a stain.
It has multiple purposes then in the agriculture industry, from a stain and also as a trace mineral provident for synthetic fertilizer. Okay.
Correct.
Thank you, sir. Sorry about that. Something.
Oh, no, it's perfectly okay. Perfectly okay. Yeah. So where are we now? Nitrogen tracking. Okay. So I talked about fixing nitrogen and transferring it to the plants. And it wouldn't be correct not to mention that we actually did experiments to confirm nitrogen transfer to plants. And, we did some of these experiments. There's plenty more that need to be done. And when we do these experiments, what we do is we use 15N, which is in the atmosphere, you have both 14N and 15N isotopic forms of nitrogen, right? So they're kind of equally mixed in the air. But in these tracking experiments, what we do is we will give plants a lot of 15N too, a lot of this isotope number 15. And what we do basically is we put nitrogen 15N in a chamber with the plant, or we just put them in air and then we'll analyze the amount of nitrogen. There's a lab in Georgia, that isotopic analysis lab, ecology lab, and we'll send material to there to be analyzed so we can look and see what happens. But anyways, we grow them for 7 days in a chamber with the 15 elevated isotopic nitrogen, and then we'll sacrifice the plant, break pieces off, and dry them and send them for analysis. So we'll find out how much nitrogen is being fixed into these tissues. Okay, so this actually shows these different kinds of symbiosis. Okay, so here we see grasses, and the size of the bar tells how much nitrogen gets assimilated into these grass leaves here. Okay, so it was this much and the grass, has its thing. And then we have hosta. We have more bacteria in the leaves because they're variegated, right? Maybe it's more efficient, I don't know, but you get more nitrogen 15 accumulating. We have our tree of heaven, which has these real aggressive plant with these filamentous trichomes, and you get more nitrogen acquired there. But then you have hops, okay, which has these glandular trichomes with chemicals in it, this antioxidant chemistry. And here you see, many times what you see in, say, for example, the grasses or the others, several times. So these are the most efficient. These glandular trichomes are the most efficient. We think they're the most efficient type of nitrogen fixing endosymbiosis in plants, and they happen to be in dicots. So you can see as plants evolve, they got more and more sophisticated. These grasses being, somewhere on the bottom, and increasing sophistication here with hops, that kind of dicot, hops and marijuana being high up on the evolutionary totem pole. And you see the trichomes now have these chemicals in them, so they're more advanced. And we can see that these trichomes, the glandular trichomes, also stain more densely for nitrogen. So they're probably accumulating more nitrogen. And this is what we're seeing in this type of experiment.
Is the staining, is that caused from the oxidation of the aluminum, or why is it blue? Is it from the oxidation process?
Yeah.
Superoxidants, right?
Yeah. That's an oxidation. Yeah, these stains, oftentimes they're oxidative. Now you're asking me hard questions. You guys are asking really hard questions.
Well, it's just my mindset. I'm trying to visualize.
No, you're absolutely right. This is chemistry. This is the ammonium molybdate chemistry, right? That's going on. It is the combining with ammonium molybdate. In this case, ammonium molybdate, that's for ethylene, but we use for nitrate detection, we use another chemical. Actually, it's the chemical we use for that is diphenylamine. And in that case, if there's nitrate present, that diphenylamine will become oxidized or reduced. I'm not sure exactly which, and then it'll change color. So yeah, this has to do with the oxidation chemistry for those stains. And we could find that. You could probably find that. We published a paper early this year on this histochemistry of bacteria and roots, and you might find that in some of those papers. But anyways, we think that we're seeing kind of an evolutionary phylogenetic signal when we studied these trichomes and how nitrogen fixation is happening, and that the nuclear symbiosis is the most primitive, and that evolved into in some dicots, this pitted filamentous trichome. There's no chemistry there, just a lot of microbes. A lot of microbes being placed throughout the structure of the filament. And then you have the glandular trichomes with their antioxidant chemistry as being the most advanced. That's what we think we're seeing. And basically that's everything. And
I don't want to end.
Well, basically, I made my case that plants are microbial processing machines, right? They are controlling these microbes. So it is to our advantage to provide plants with microbes so that they can do the thing that they do, get nutrients from those microbes, and that our crops will be better off, we'll be healthier, the environment will be better, and, regenerative agriculture, this supports regenerative agriculture. And, the plants need these microbes for proper development and fewer chemicals. So, the questions, they're all right there. Some questions, like, for example, I guess, have we damaged? One question, for example, have we damaged in our crops all of the microbial systems? Have we damaged them over time by using fertilizers or antifungals or any kind of, sterilants? Have we damaged them? Can we get them back? I mean, if we've damaged them, can they get them off from the soil, from a healthy soil, if we cultivate them there? Can we go back to for example, wild relatives of plants and get the microbes off of there and then bring them in and put them on our crop plants and reacquire these systems? Okay, so another idea, and I think we've already alluded to this. Can you cultivate crops just using the microbes? Can we use microbes to control disease, to give fertilizers, to make plants hardier, and so forth? Can we develop the knowledge where we know how, and the products too, where we know how to put microbes for specific purposes in specific places? Another question is, how can we support these kind of endosymbiosis in plants? There's this whole concept of foliar fertilization, right? Well, foliar fertilization, people do it with teas, they do it with chemicals. Sometimes they put chemicals on leaves instead of on the soil. Can we do a kind of fertilized plants using variants of foliar fertilization that support these endosymbioses in the leaves of plants, right? Can we give, for example, could we give them molybdenum and magnesium and iron maybe, because those are the metals, the nutrients that the microbes need to fix nitrogen in the leaves? Could we just give them the raw ingredients for nitrogen fixation and then have them, so we don't have to put any nitrogen fertilizer on the soil, we just put some cofactors, some metals on the leaves, and then the bacteria in the leaves can do all the nitrogen fixation. So we can push that process so that we have plants then growing, making all their own nitrogen. So those are some of the questions. You could think of a lot of other questions too, and have some other ideas.
Over the last 10 years, 12 years now, I've put most of my dedicated energy into actually one area that I think will make the most significant. As I read everyone here, the main problem, I hear it as being toxic water that makes the microbes dormant. And so I've spent a lot of time trying to clean up the water to create a healthy environment for the microbes to get reestablished. Obviously, not over fertilizing, but all those over fertilizing is just incomplete man-made chemical reactions, so which are toxic. The same thing of what we're discussing. So, yeah, everything you're saying, I always just think, what is that one thing that it really inhibits the microbes from taking off? Is clean water and all the stuff we put in that water that ends up throwing off all the chemical reactions in their environment. Do you agree with that or disagree with that?
Well, I honestly think that there's a lot of things that we need to do to support microbes in soil, right? Not putting, say, water that has detergents in it, for example, right? Or not having excess, phosphate, because that'll screw up the microbial balance in the soil too, or, whatever it is. And that goes along with your toxic water idea. Yeah, that's one thing, right? But the other things relate, for example, to having organic material in the soil so that these microbes can live off of that while they're not actually in the plant rhizosphere or in the plant structure itself. So having this organic base that these microbes can survive in. So that's important, right? Having oxygen in the soil so that plants in the root hairs and plant roots growing in that soil have the soil oxygen that they need in order to produce superoxide and extract nutrients from those microbes. So if you have a hard pan, if you're driving over the soil constantly and you made a hard pan down there, you have areas where then soil becomes flooded and there's no oxygen there, then you're going to inhibit this process. You'll have water there, but you'll not have the oxygen in there that plants need in order to extract nutrients from the microbes. So I think, water is one aspect, having water that's not contaminated with stuff, having soils that aren't contaminated, but also, your environment of oxygen environment and organic environment, are all really important in soils. And really, when you're creating a regenerative soil, you're trying to remediate all those things, right? And your populations, you want your population. Typically, the soil microbes will build up if you have the right environment for them, with organics in there, and you have typically there's more involved than just the soil. You also have to have cover crops where you keep those microbes alive when you're not growing crops there, because you need some plants there to feed them other nutrients, and so you keep those populations healthy. So it takes multiple factors.
Yeah. And that's we were just talking about, we just filmed a video the other day, and I was explaining that, like, for me, people that are entering organics or wanting to further, I look at my nutrients, not just food sources for my plants, but in the bacteria, but also contributing to the overall integrity and the structural integrity of that soil, allowing that aeration and stuff. So things that might be a little bit larger that we're putting in our vegetable gardens, like oyster shells or larger matter, it takes longer to break down, but it's also contributing to that aeration and that breaking down and that further process for food sources and allowing proper oxidization and especially drainage as well too. So that's just kind of something we were just recently even talking about, and that's my mindset too, where if I'm trying to direct people to creating better soils in their gardens and their plants, their medicinals or whatever in-house, this is part of the process, that we have to have all these variables and not just say, oh, well, it has 35% calcium, but it makes up so much more of it in my mind. That's how I look at it. And that's how I'm trying to kind of help people.
Soil is a living thing, right? It's not like we just need to put a mix of chemicals there. It's a living structure. Soil is alive, and we need that life in order to grow plants. Plants rely on that life being there. And if you take it away, if we take that away, we know, because we can do it in the laboratory, we can take all that life away, or much of that life away. And when we do, plants are sick. They are sick. They're sick. They won't grow. They can't grow. The roots don't grow down. The roots don't grow root hairs. The roots don't grow much. The leaves don't grow much. The plants need those microbes. And if you take them away, plants become sick. So it's a living. Plants need a living soil. Healthy living soil. Yeah.
James, man, if you were my teacher, I probably would have never dropped out of college one of the 4 times. I'm serious, man. I got bored. And I was just like, all right, I feel like I feel so slow. This is what I search for on my higher education now in my life. All right, guys, only on Perfect Gardens TV. Remember to please like, share, and subscribe. I normally recommend for them to grow out all the strains they're growing. Keep the 1, maybe 2, but normally 1 strain that they absolutely love, and get rid of all the rest. And to start to learn how to read the plant through plant deficiencies,